Great. So it's great to be here today. It's my first visit to Houston. I've flown through here many times, but never actually visited here, so it's great to actually visit this university. So I'm going to give, this is an overview of the talk. I think I want just a couple slides on what NIBR is, where I work now, a couple slides on that so you're familiar with that institution, and then I'm going to talk about some of the paradigms and technologies, particularly on low molecular weight screening. This is my main area of expertise. I'm not going to be able to cover everything, but I'm happy to talk about biologics in drug discovery as well, perhaps during the question and answer period. And then give a couple examples of how we can improve hit-to-lead processes, improving assay design, introduce this new platform at Novartis called Microcycle version 1, and I'll describe what that is. And then the paradigm we're using to actually work with academic collaborations in my lab. So the Novartis Institute for Biomedical Research is the research arm of Novartis. Novartis is a huge global company of over 100,000 scientists. Novartis Institute of Biomedical Research is about 6,000 scientists worldwide, with headquarters in Cambridge, Massachusetts. There's about a couple thousand scientists working in Cambridge. The bulk of many others are actually the headquarters in Basel, Switzerland, as well as GNF in San Diego and Emeryville, which houses our tropical disease unit as well. Currently covering about 400 different projects, covering about 90 molecular entities. And this is organized around several platform groups and disease areas. They're illustrated here. The platform group I'm involved with is chemical biology and therapeutics, which provides all the kind of high-throughput biology, resources for discovery biology, as well as target identification, chemical genomic type and genetic screening approaches, as well as certain disease areas as well are integrated within the chemical biology therapeutics platform. We also have the new Biologics Center, Global Discovery Chemistry, and Translational Medicine, which all work with the disease area platforms. I would say immuno-oncology has grown a lot in the last few years, and this has impacted kind of how we think about screening in a bit. As well, we brought in neuroscience back actually a few years ago with really focusing on Alzheimer's disease and autism. Infectious disease actually is no longer, our disease, we have a tropical disease department, but infectious disease is actually not a part of Novartis' portfolio as of about a year ago. So one view on chemical biology is just finding what fits. And of course, we're all familiar with some of these general modes of trying to perturb proteins or particular biomolecules, either orthosteric or allosteric modulation, or perhaps through a biologic, through a ligand trap. Some of the newer modalities which are focused are things like targeted protein degradation, and here we have a compound binding, recruiting an E3 ligase, specifically targeting this GFP expression protein, but not a non-target for that compound, which is fused to the red fluorescent protein. And this is really based on the finding of thalidomide being able to essentially act like a molecular glue, changing the conformation of an E3 ligase like cereblon, and bringing in a new target and selectively inducing its degradation. You can imagine other modalities which might be useful with this type of mode. And they could include this type of binding as well, where you have an archetypic type binding, a hallmark of some GTPases for RF-sec interaction, where the compound has no affinity for either target, but in the context of a complex, it can form a very tight ternary complex in a kind of an induced fit type way. So this is, these types of new modalities are some of the we're thinking about biology that's been tough to drug in the past. Another view of chemical biology is with respect to the compounds that we screen. And I'll have more to say about this in a minute, but this is one kind of why I view the spectrum of different types of compounds. They range from tools, probes, to drugs. Drugs, of course, are being the most highly sought after molecules, but very few compounds achieve that, the criteria to be safe and efficacious in man. However, we've found a lot of useful tool compounds throughout the history, including things like cyclohexamide for studying protein translation, doxycycline, which can be used not only as an antibiotic, but also to make inducible assay systems. Of course, things like forskolin, natural product, extremely useful for developing GPCR assays. And the use of through the many different academic efforts and the use of like the molecular library screening network, many different highly quality chemical probes have now been developed. And these are really characterized by being very specific for a particular target, and usually we want to have good cell penetration, cellular activity associated with them, and of course selectivity. So they're kind of a specialized, kind of fit for purpose, has been called, type of compounds. And of course drugs themselves can be useful to study biology, and of course they're also extremely useful for, the main use for treating disease, but maybe not so useful for probes. For example, erythrochromic acid, which is a diuretic, has a Michael acceptor covalent modifier. It's not going to be as useful for studying like broadly biology. Or they may be a prodrug requiring in vivo metabolism to actually become active, so that the actual parent molecule is not active against the target. They can be useful probes as well, like Citadel is a great PD5 specific probe. Or this one here, which is really just an adenosine analog, can be somewhat non-specific, but is a useful chemotherapy agent. And so I think one of the paradigms that has come out over the last more than 10 years or so is that historically, at least when I started, we would screen very large libraries in a pharmacopoeia. We were combi companies, so we had libraries over 7 million compounds screened in mixtures. So very, very large libraries. And we would use kind of uncharacterized biology to try to understand the activity of the type of compounds that affect different types of biology. But now over time, we have that database available to us, and historically, like Novartis has screened hundreds of assays, and we can look at high throughput fingerprints with respect to technology or particular biological target class, as well as where we have IC50 or KD information for a specific target, like a kinase, GPCR, or a protease. And we can use that information to design both chemically diverse and biodiverse libraries. So now this has actually kind of turned it on its head, where now we use the compound library to characterize a bioassay as opposed to the other way around. And so actually, one of the results of this type of thinking is that it's been relatively rare at Novartis where we would screen, in the last few years, I'd say a full deck, 1.2 million compound library. Most of the time, what we screen are very small collections of, say, 10K, 50K, up to 250K screens, which are these biodiverse and chemically diverse libraries. So this is actually kind of a shifting paradigm. And I think there are two reasons for that. One is just that, we have gone into areas of biology which are pretty high risk. So we don't know a lot about what type of chemical matter might be useful, or what type of assay to use to actually address that biology. So we want to run pilot screens of, say, 50K compounds, 10K compounds with these highly annotated collections before we go to committing to 1.2 million compound screens, getting hits out of that screen, and then not really knowing, if they're going to translate downstream or not. So this is a kind of a shifting paradigm, I think, which is not, I think, not just true for Novartis, but from other industry companies, I think many people are actually going this way. So there's been less, I would say, large compound screens and more focused screens as a result of understanding of the behavior of our libraries. And a special case here is a library which has been developed over, I'd say, the last 10 years through cheminformatic scientists at Novartis, as well as chemical biologists who are nominating, well-validated chemical probes for particular targets, and also just judging and scoring compounds as well as they behave in their hands. And we can use this to construct this library called the Mechanisms of Action library. It's about almost 5,000 compounds. The composition of this library, we hope to be out in publication by the end of this year. So we have a manuscript and review. Hopefully it'll get published soon, and we will publish the whole contents along with the compound structures, as well as the primary target ID and the methods for actually deriving those compound target interactions. But this is very useful for like phenotypic assays. For example, if you have a phenotypic readout, you're not really sure the type of biology that's affecting it, and look at the types of targets that come out. And usually the way teams use this is sometimes they identify mechanisms of actions that they don't want. For example, they might find that their particular biology is very sensitive to HDAC inhibitors, and that may be something not of interest to them, so they want to include an HDAC binding assay in their flowchart downstream. Or through pathway analysis, by looking at the polypharmacology of the compounds and the target annotations, you might be able to come up with a particular pathway hypothesis, which you can then go further explore with additional compounds annotated to that pathway or with genetic tools. And that can probably, many times, often leads to a different assay or different phenotypic assay readout that's more relevant to the type of biology you're interested in. So this is a very useful library for just characterizing those types of phenotypic assays. And because we do so much of these pilot screens, these 50K type screens, as I mentioned, of course, we're a local company, but they have these very nice modular type based systems where you essentially wheel up carts. The other systems, which are in great demand, is the so-called big yellow arms, or BYA systems. Nice thing about these GNF dispensers, so these are actually very flexible systems now. Another area of, I would say, growth, we have systems similar to the rapid fire systems, so we can do roughly 10 seconds per sample or something on that order. We have 4 different injectors, and it gives us the ability to do like 250K screens, compound screens with an LC-MS type detection. Another one which we've implemented in the last few years is a MALDI-based detection system for looking at peptides or protein type product reads. In this case, we use a TTP LabTech Mosquito to actually dispense nanoliter quantities onto a MALDI plate that goes into the reader, and this gives us a throughput of about 15,000 samples per day. So you can do up to 50K screens with this over a period of a couple of weeks. Of course, the other thing is high content imaging. The high throughput confocal microscopes have gotten extremely robust. And given all that high content imaging data, we've really tried to implement robust informatic pipelines to make use of all the information that comes out. So rather than just looking at what you think is going to be interesting for that biology, intensity readout or cell number, look at all the parameters you can get out of the image, texture parameters, as well as intensity and morphology, and then use statistics to try to figure out which parameters are actually being useful to define the type of biology of interest based on, hopefully if you have positive controls, you can do this type of work. As an example of that, here's a relatively simple assay where we were just looking at maintaining pluripotency of a human embryonic stem cell line. So this cell line basically has a GFP reporter in it. It's an embryonic stem cell line engineered with a GFP reporter, where if you add doxycycline, you induce some transgenes, which drive it into the so-called naive state of pluripotency, which is the desired state to start with when making induced pluripotent stem cell type derived lines, because there's no particular bias towards any type of cell type at that point. And when you remove dox, the cells become less bright, and they start to go into this prime state and differentiate. So certainly you can see a change in brightness of GFP in the positive and negative controls, plus or minus dox. But you also may note a big change in morphology. So these are in the pluripotent naive state, they have these kind of round dome-shaped colonies associated with the phenotype, where here they're very flat, spread out cells. So if you just look at GFP intensity, you might score something like this as active, but of course this is nothing like the positive control. This turns out to be an HDAC inhibitor, which is just non-specifically upregulating GFP expression. But if you use a homogeneous distance that includes both GFP intensity parameters, as well as things like shape, morphology, compactness, for example, you can now score compounds like this, which are much closer to the active control and actually do have a phenotype which looks something like the positive control, obviously not as strong, but they are round, bright GFP colonies, as opposed to these things which are very small, kind of dead cells and spread, and also have many kind of debris associated with. This is probably a toxicity effect. So I think even with a relatively simple readout like this, you can learn a lot and get a lot better scoring using multiparametric data analysis. So this is something we've implemented on the Amazon cloud internally, because it does take a lot of computational resources, but it's well utilized for high content assays. So now I'll talk a little bit about improving assay design interpretation. This is just a depiction of what the simplest view of an assay that I could think of, where basically you have something you want to measure, a pathway or a target, and then you have to develop a light signal, because usually we have to measure some sort of light emission associated to get a detectable assay, and then you make a measurement. And so the important thing to remember is a lot of things happens between target and measurement, and that X here is what interferes with a lot of the interpretation. That's what a lot of my work has actually been studying, what things that interfere with assay technologies. Really starting around 2004, we started working with the scientists at Eli Lilly who had an internal document called their Quantitative Biology Manual. This is a document that had been existing at Eli Lilly for more than 15 years. And they were willing to share that manual with us in 2004, and we started to make it available publicly for free. And ever since then, we've been adding chapters to this. So there's been probably 50 or so scientists from industry and academia which would contribute chapter to this now, with more than 43 chapters. They are peer-reviewed by industry experts, and we try to make this kind of like the Bible of how to develop good assays. Okay, so here's a simple assay, one that I've studied quite a bit. We were simply looking at transcription of that reporter driving through a promoter, specific promoter, so you're looking at some sort of pathway stimulus that affects that expression. And they're quite useful for miniaturized assays because you simply seed cells, incubate with compounds, usually overnight or so, and then add detection reagents and read. So they can be quite useful for high throughput and some related shorter half-life type luciferases. And this is a result of my screen of the PubChem library. When I was at NIH, we screened the PubChem library of 350,000 compounds, actually in dose response at 8 concentrations. And at Novartis, I screened the full 1.3 million compound file at 10 micromolar. And both results similar results were derived from each in that firefly luciferase compose about 4% of typical screening libraries. Actually, what happens when you run these types of assays and you pull in all those hits, your hit list can be enriched by 40 to 98% with these type of F-loop-based inhibitors. And we also found a counterintuitive artifact associated with this, which was actually quite confounding at first when we saw it, but once we understood it, it made a lot of sense, in that many of these inhibitors in cellular assay systems actually show an activation response. So here's this benzthiazole compound, which looks a lot like the D-luciferin substrate. This is the activity against the biochemical enzyme assay, so it's highly potent against the enzyme. In the cellular system, you see this kind of activation, which can range from a bell-shaped curve to a sigmoidal curve. As it turns out, these compounds, like many compounds that bind to particular targets, stabilize the enzyme structure in a way that's concordant with the IC50 against the enzyme. Some of these show delta Tm values of more than 10 degrees. And so what's happening in these cellular assay systems is that when you treat the cells with compound, the half-life of that reporter increases relative to the untreated wells. And then when you add detection reagent, many of these inhibitors are competitive with substrates. So the inhibitor falls off the enzyme and you detect near full activity. And then you measure that as something that looks like gene activation, but actually has nothing to do with gene activation. It's just the increased half-life of the inhibitor-bound enzyme in cells, including firefly luciferase, vanilla luciferase, and some variants which show either red-shifted type luminescence. Most luciferases are blue or green, but there's been some variants that are out showing red luminescence. They may be ATP dependent or non-ATP dependent, taking solantracine or D-luciferin, for example. Included some of the new luciferases like nanoluciferase. It's called nano. It's developed by Promega because it's at that time the smallest luciferase known, about 9 kilodaltons. Again, these look like specific enzyme inhibitors and not something that's just doing something non-specific to the particular luminescent signal. If you look at these compounds, a lot of them are specific N-look or F-look inhibitors. So just by looking at a single well from the primary screen, you can already reduce your hit rate by about 50% and get some and prioritize compounds in this region here, which can greatly help your follow-up study. So this is a solution to the problem. So this could be a nice alternative as well for a single reporter. You could use turboluciferase. It's very small, 16 kilodaltons, shows glow type response, and appears to be a tough target. The reasons for this were not completely understood, but it is kind of unique in that it does bind to substrate and involves cooperative substrate binding, which is different than any of the other luciferases. So maybe that plays into its ability to be inhibited by, inhibitor binding to a single site is not enough to lead to a reduction in luminescence. So this is another one to keep in mind. It's from Thermo Fisher. So scientists need time to innovate, to try to address the bottlenecks. Where things get very complicated is you're trying to optimize many different parameters at once, ADME parameters as well as potency and solubility, for example. We're getting very good at hit generation through the processes I just mentioned, thousands of hits that are high affinity. But how do we choose which hits to actually work on? So we wanted to bring these all into one lab in an integrated system enabled by lab automation. So we're past the 18 months now. It's been implemented. We call this a GPS for drug discovery during our pitch. This type of model was used for those investigators to come into our lab. They would usually bring an associate from their lab to work with us. We would have an automation expert, assay development expert, work with them to help optimize the assay, whatever they needed to do. Because they're asking pretty succinct questions, the actual projects themselves would last maybe a couple of weeks to a few months, so they're not like multi-year projects, but kind of come in, run the test, get your data back, and then move on. A lot of them were low molecular weight screens, looking for tool compounds. We also did some genetic screens. We would provide all the same resources that we provide internally, as well as compound libraries to academic investigators who had something that looked of interest to us, but was different than we were currently working on. And so the Novartis Academic Fast Lab Agreement, there's no money transferred, it's an in-kind. Each side puts resources in. But basically the main thing about the agreement is that it has language about what will be brought into Novartis, what reagents or assays are being brought into Novartis, and also visiting scientist provisions, because usually a postdoc or graduate student will come work with us for a few weeks. Usually it's about 90 days of work, different visits, through different visits, in order to complete the work. And there's no IP, so we're actually not free to work on anything that we find out of the collaboration, but the investigator gets all the compound data, screening data back, and they're free to do whatever they want with that information. Of course, we do encourage publication. This is often the goal, because many times this involves a postdoc. So a lot of times co-publications do come out of this as well. And what we get out of it, I think, is we get a chance to look at a new assay approach or new technology, something we wouldn't normally have the time to look at, and just evaluate how it works in an industrial setting. Or it might be a new target class that we don't have, hadn't really put any emphasis on yet or thought about. Just gives us the ability to see what that particular assay, that approach can be useful for that new target class. So it gives us some information about that, but it can't be something that we're already working on. Obviously, that would go to a different type of agreement. These are actually something we're not working on, but it looks interesting. So it's in that kind of special, kind of exploratory space. And the research plan is really just a one-pager with a little background information, a statement about what will be screened. I'll talk about the libraries here in a minute. The type of assays that will be brought in, again, who's visiting and who's doing what in terms of following up on the compounds, and then what do we expect the outcomes to be for each side. So the compound libraries come in a couple different flavors. One, the largest collection is a diversity set, which is based on the same rules I mentioned that we use to design our internal set. So about chemical diversity, high chemical diversity and biodiversity is actually a subset of our full deck library. It just happens to be compounds that are in the public domain. So these are compounds we actually routinely screen internally, but we plated them out separately for this purpose. The initial compound set can be looked at as either a core set. If you have a really difficult assay, you can just screen the 7K and then do hit expansion from a 40K set. But recently now, we have approval for an additional 160,000 compounds from the Novartis deck, which can be used for hit expansion purposes. So those 160 are not plated. But once you complete one of these screens, you can cherry pick, do SAR by archive, and try to get additional SAR information from that 160K screen. The other, that's just a depiction of the larger library. Again, good chemical properties. But the other library we have is a purified natural product set. This is about 2,500 natural products. So Novartis has always embraced natural product drug discovery. There's a big unit in Switzerland, which is continuing to characterize these. These are purified natural products which we can share. So this gets you into a different area of chemical space. And again, these compounds we can reveal all the structures and any of the data that comes with them. And then, of course, the other library we have, which will show a little example at the end, is the mechanism of action library, which is publicly available. So that's another for cellular assays. And there's an example of an early one we did with Eric Kuhl's lab at Stanford. He was introduced to this enzyme called oxidoguanine glycosylase, OGG1, which is a target in cancer. Activation of this enzyme is thought to prevent cancer lesions, whereas inhibition may sensitize to various types of radiation treatment, for example. So actually activators or inhibitors could be important, but it cleaves 8-oxidoguanine from DNA, which is one of the most prominent forms of oxidative damage that occurs in cancer cells. And what the particular reagent what he had developed, that we were interested in, was this prophorescent oligonucleotide substrate, where you have a quenched 8-oxidoguanine residue with this prophorescent substrate. When OGG1 cleaves and you form this A basic site, you actually get emissive of the luminescence, of the blue type luminescence. And so this is a unique substrate to look at this DNA RNA pair enzyme. It's a new class of enzymes for us, which we hadn't looked at. We were also interested in oligonucleotide potential compounds that might bind oligonucleotides. That was something else we thought we could maybe learn from this assay. So it's an interesting assay system for us to look at. And we ran about 50K compounds with one of his postdocs. We found this compound here, this tetrahydroquinoline compound, which Eric Kuhl's lab, who has the ability to do synthetic chemistry, made several tens of analogs and drove the potency from a few micromolar down to 60 nanomolar. And this is some SBR data we collected, which shows that we get a KD concordant with the IC50 against the enzyme of about 100 nanomolar. So it binds to OGG1 alone, not in the absence of DNA, so it doesn't actually bind to the DNA oligonucleotide. It is an inhibitor. We didn't find any activators in the screen, but we found some potent inhibitors such as this, which this nicely appears to be selective against a number of other DNA RNA repair enzymes, and also showed a reduction of ozonic guanine lesions in HeLa cells, so appeared to be cellular active. So this was published in JACS last year. I think it's a nice sample of a new chemical probe for this target class, which we were able to work on. And then the last example is more of a phenotypic approach, and this was done with Lori Boyer's lab at MIT. Her lab is broadly interested in treating heart disease for studying the maintenance and proliferation of cardiomyocytes. So these are cardiac muscle cells, which in normal adults don't really proliferate at all. They have less than 1% annual turnover rate, so during cardiac arrest, these cells are lost and never really can be regenerated. So finding ways that you can efficiently regenerate the cell could be very useful for treating heart disease. And her model system was to use neonatal mouse cardiomyocytes. So that provides a system where we could potentially perturb it to look for things that would increase the proliferation rate. Normally after about a week, they would exit the cell site and become binucleated and turn into just senescent cardiomyocyte cells. So we used P1 cardiomyocytes from P1 pups. In the literature, there's very little known about what regulates this, although P38 and GSK3 betas are thought to be negative regulators of proliferation. So inhibitors of these have been described, which actually shows some increase in proliferation rate, and those could serve as positive controls. So you use the mechanism of action library exclusively for this program. We were dealing with primary mouse cells, so we couldn't get a lot of cells, but we had enough to do several different rounds at different concentrations of this small box library of about 3,000 compounds. And the workflow was that the postdoc at MIT would harvest the cells, bring them over, we would plate them in 3D 4-well plates on day one, and then treat them with the mechanism of action libraries, and then stain for proliferation starting at day 3 with EDU. And then after fixing, we would stain with Ki67 as well as DAPI, and to define cardiomyocyte cells, troponin T, the main muscle component of cardiomyocytes, and use high content confocal imaging. Nicely, when these cells are plated, they actually do beat. So that's and actually quite rapidly, which is a hallmark of young kind of cardiomyocyte muscle cells. So they appear to be healthy in those plates. And then for the image analysis, the main trick was that because these were primary cells, we did have some contaminating cardiac fibroblasts. So we had to distinguish fibroblasts from cardiomyocytes, and of course we could do that with our troponin T stain. Fibroblasts would be TNT negative, whereas cardiomyocytes would be TNT positive. And then within that green area, we would then enumerate all the cells which show troponin T, and then look for increase of the proliferation marker Ki67ADU. But the internal fibroblasts, which are present in every well, provided a nice internal counter screen because we really wanted compounds that affect the proliferation of cardiomyocyte and not just any cell. So we could then look within the same well, are we only seeing proliferation index increase of cardiomyocyte and not fibroblasts plus cardiomyocytes? However, we did find novel target classes as well, looking at all the different target annotations. One of the prominent ones that showed up were a bunch of calcium channel blockers. So we found most of the calcium channel blockers in our library. These are 3 different chemotypes that were associated with this, but they were all annotated as calcium channel blockers. And they actually scored quite well in the assay, showing pretty strong proliferation relative to the chiron control for the cardiomyocyte population, but really nothing against the fibroblasts, as an example of one we showed up. And that was a novel type of class of inhibitors which had not been described before. It does make some sense because these are thought to be cardioprotective in many studies. So these are now being followed up at MIT by RNA-seq to try to better understand the pathways that the various compounds we validated actually, what type of pathways they might be affecting. So we've worked with more than 30 different institutes over the last 3 years, and there's now a lab in both Basel, Switzerland, as well as in Cambridge doing this type of work. This is an example of some of the places we've worked with. So they're not all local to Massachusetts. We have people coming from as far away as Brazil. We had 2 investigators come this summer from Brazil, which were hosted by the Tropical Disease Unit, looking at enzymes from T. cruzi, for example. So again, if you're interested in finding more about this, I can talk to you later more, but this, my email address is here, but this has been a viable program which we continue to support for low molecular weight screening with academic labs. And that's all I really wanted to say. These are the people that were involved with some of the reported gene assays that included work at both Novartis and NCATS, Thermo Fisher and Promega, and many different people that have been involved with the FAST lab making this really viable as a nice model for us to work with external collaborators. So, thank you.